Carbohydrates. The definition simply means that it contains carbonyl compounds such as aldehydes and the ketones. It consists of carbon, oxygen, hydrogen atoms with hydrogen-oxygen atom ratio of 2:1. The empirical formula will be this. Carbohydrates are classified into monosaccharides. These are simpler sugars of empirical formula C6H12 and O6. These cannot be hydrolyzed into simpler carbohydrates. Examples fructose and glucose. The next comes the oligosaccharides. This consists of 2 to 10 units of monosaccharide units. Disaccharides means the condensation of two monosaccharides units. Glucose and glucose gives maltose. Fructose and glucose gives sucrose. Galactose and glucose trehalose gives lactose. This consists of a glycosidic bond linkage. The next comes the polysaccharide. This is also called as glycans. This contains more than 10 monosaccharide units and they can be hundreds of sugar units in length. There are two types again, homopolysaccharides, single type of monosaccharides. Heteropolysaccharides, polysaccharide molecule consisting of more than a single kind of monosaccharides. Example, hyaluronic acid, heparin and chondroitin sulfate. So these are some of the examples of the carbohydrates which are cellulose, starch, glycogen and chitin. They have a glycosidic bond linkage between them. Carbohydrates are divided into simple and complex. Simple are further divided into monosaccharides and disaccharides. So complex examples are starch, glycogen and fiber. Monosaccharides example are glucose, fructose and galactose. Disaccharides examples are maltose, sucrose and lactose. So digestion of the carbohydrate takes place in three places. First in the mouth. Hemolytic enzyme present in the saliva is salivary amylase or ptyalin. In stomach, the gastric juice contains the weak amylase. In the intestine, the amylolytic enzyme present in the small intestine derived from the pancreatic juice that contains pancreatic amylase, and the succus centricus that contains the maltase, sucrase, lactase, dextrinase, trihalase. Example. Absorption. Absorbed from the small intestine mainly as monosaccharides, namely glucose, galactose and fructose. Energy obtained from the binding process of sodium ion and glucose molecule to carry a protein. Glucose gets absorbed into portal vein by facilitated diffusion. From the intercellular space, it moves to the blood by active transportation. And this gets breakdown into ATP and this gets breakdown into energy. Metabolism, the process in which food substances undergo chemical and energy transformation. Catabolism simply means the utilization by oxidative process in which carbohydrates, proteins, and lipids are burned slowly to release the energy. Anabolism, released energy utilized by the tissues for physiological actions and rest of the energy is stored as energy-rich phosphate bonds and in the form of proteins, carbohydrates, lipids in the tissues. So there are two pathways in which the glucose enters inside the plasma membrane. There are two pathways in which the glucose enters and it may also get exited out. In order to prevent this, the phosphate molecule get attached to the glucose molecules. After this step, the ATP gets converted into ADP with the help of hexokinase enzyme. The glucose gets converted into glucose 6-phosphate, in which the ATP gets converted into ADP with the help of hexokinase enzyme. Fructose 6-phosphate is a reversible reaction in which it gets converted into glucose 6-phosphate as well. With the help of isomerase enzyme, fructose 6-phosphate is formed. And fructose 1,6 bisphosphate is also formed with the help of phosphofructokinase 1 enzyme, in which the ATP gets converted into ADP. This phase is called as energy investment phase as the ATP is lost. After the formation of the fructose 1,6 bisphosphate, in which the phosphate group attaches to the 1 and the 6th place in the carbon chain, which is then converted into glyceraldehyde 3-phosphate which is an isomerase of dihydroxyacetone phosphate, which is shortly called as GAP and the DAP. Only the glyceraldehyde 3-phosphate goes into the direct pathway in which the dihydroxyacetone phosphates gets converted into glyceraldehyde 3-phosphate. Now we shall see the function of the dehydrogenase enzyme, in which the oxidation reduction reactions takes place. At first there is a molecule in which the hydrogen atom is attached to it, and then another molecule, and in the product the simpler molecules gets attached to the hydrogen and then another product is formed. In this way hydrogen gets replaced to another molecule, which with the help of the dehydrogenase enzyme. The glyceraldehyde 3-phosphate in the presence of water gets converted into high intermediate energy molecule, in which the phosphate gets separated and forms the 1,3 bisphosphoglycerate. There are two molecules, that is the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. So this glyceraldehyde 3-phosphate gets converted into 1,3 bisphosphoglycerate, in which two NADH molecules, NAD molecules gets converted into two NADH molecules with the help of the dehydrogenase enzyme, that is the glyceraldehyde 3-phosphate dehydrogenase enzyme. So there is a substrate level phosphorylation taking place, that is two molecules when binds produces energy. Then the 1,3 bisphosphoglyceride gets converted into 3-phosphoglyceride with the help of phosphoglycerate kinase, in which the ADP gets converted into ATP. The first energy producing step of the glycolysis is this step. The 3-phosphoglycerase gets converted into 2-phosphoglycerate with the help of the enzyme called phosphoglycerate mutase. And then this gets converted into phosphoenol pyruvate with the help of enolase enzyme. Now this gets converted into phosphoenol pyruvate, in which the 2 ADP gets converted into 2 ATP. And the end product is called the pyruvate, in which it is converted by pyruvate kinase enzyme. Now we shall see the energetics of the glycolysis. During the anaerobic glycolysis, during the absence of the oxygen, there is a 2 ATPs used in investment phase and 4 ATPs made in the payoff phase, in which results in the 2 ATP gain. And in the aerobic glycolysis, during the presence of the oxygen, there are 2 ATPs used in the investment phase and 4 ATPs made in the payoff phase and 2 NADH plus used in the electron transport chain, in which there is a results in 2 ATPs and 2 NADH. Regulation of glycolysis occurs in the nucleotide synthesis, lipid synthesis, and Krebs cycles, electron transport chain. So these are all the pathways in which these glycolysis takes place. Thank you.